Okay. Now let's go to another server. Let me just go to the PowerPoint slide. So this was the server that we just discussed now, Epitopia, where we entered the PDB ID and chain again, the ID that we got from the BLASTP, right? Yeah. Now the next server that we'll be going is SEPA3 to predict the glycoprotein antigen epitopes. So for this, go to this particular link. Or Google SEPA3 and go to the second result, where you'll be redirected to the SEPA3 homepage. Then here what you need to do is, you need to click the start button. So click start. Next you need to enter the PDB ID and chain. Then you need to give your mail ID and click submit option. That's what you need to do. So let's enter the PDB ID, which is 4XAK. Then the chain, it was A. Okay. Then you need to subcellular. Okay, that's all. Then make everything default. Give your mail ID and click the submit button. By the time results are ready, let's come back to the Epitopia results. So the results that I gave for 4XAK, it has now received, where we can actually, again, let's just visualize that particular result. Okay, let's take the second result, which was the immunogenicity scores. As you can see, the highest one is 0.78 to 0.188 immunogenicity score. So the immunogenicity score is 0.782 and the probability score is mentioned, and it's all exposed. Okay, so 0.188 is the probability, immunogenicity is 0.782. So that means all these are considered to be the epitopes which are exposed ones. Now let's go to the SEPA server and see the results. Here are the results. So it's chain A, threshold mentioned was 0.089. So chain A, threshold, it's 0.089. So here we did not specify the subcellular localization, so that's why the species of host was also not specified. Anyway, that all depends on your research aim. So here, according to the results, the total number of residues were 211, but the number of predicted epitope residues under this threshold, there were 51. You can always change your threshold and see how much the number of epitopes you're gonna get. Okay. So here you can see the number of epitopes identified under a threshold, right? So here the residues marked red are actually the epitopes. Now you can see the predicted score here, and you can even download the predicted PDB file. You can download both this. You can click to see the 3D structure of it. So this is your 3D structure, and you can save this file, save it as JPEG, PNG, in all other formats. Okay, so yes. Now when you click here, you'll get the highlighted epitope residues that are predicted. See, if you do not click, it will go. So these are the highlighted epitope residues. Okay, yeah, if you want to spin on, you can spin on. If you want a cartoon model, if you want a stick model, ball and stick model, anything that you can do. So this is how you interpret your results. So what we learned today in this lecture was, again, under functional analysis, we predicted the discontinuous epitopes from the 3D structure of proteins. We then predicted immunogenic regions again in another server, and again about the we understood about the immunogenicity score, what does it mean, probability score, what does it mean, and then we predicted the glycoprotein antigen epitopes as well by looking at another server. Thank you.